thank you very much for giving me the opportunity to give this webinar. And in addition to you having the opportunity to ask me questions, I'm also going to ask you some questions. So it's important that you have your mobile ready, because you're going to have to scan some QR codes in order to be able to participate to the interactive aspect of this lecture. So before going into the lecture, some disclosures. And that is mainly because of my work in Duchenne muscular dystrophy, where I have been a co-inventor on patents which are held by LUMC, so my employer. I'm also doing a lot of advising and consulting, and all the money for that goes to the LUMC and not to me. And I will sometimes speak in company-sponsored symposia at conferences, and also that money goes to LUMC, not me. So that's just the logistic part out of the way. So we're going to start with an interactive question, because we're going to talk today about how patients can be involved in therapy development and how they should be involved in therapy development. So this is the first thing where you can scan the QR code, and then you can just type how you think that patients can be involved in therapy development. So I'll just wait for people to scan it and do it, because it should be working. So I see one participant typing. Okay, so the patient outreach groups, yes. And there's no right or wrong here. Patient-centered outcomes, yes, you need patients to develop them. Interviews, assessment criteria, so outcome measures. Ask patients what's important to them. Very a good question. We'll come back to that, so that's nice. I'll give you some more seconds to type. Samples, yes, indeed. And then let's see what the new one is. Personal inter-protocol review, etc. So most burdensome symptoms. So I see a lot, and I think one thing that we don't see yet is that they can, of course, participate to clinical trials, and that's another way that they can be involved. And they can, of course, also fund research. So there's many different ways that patients can and should be involved. So the disease that we're gonna discuss today is Duchenne muscular dystrophy. And Duchenne is a disease that primarily occurs in boys, because the gene that is mutated and the protein that's missing, the gene that's mutated is located on the X chromosome. So women will have 2 copies. So if there's a mutated copy, they have a backup. But men have only 1 X chromosome. So if there's a mutation and they cannot produce dystrophin, they will get Duchenne muscular dystrophy. And this is characterized by progressive loss of muscle function. You can see this is a very young patient. You don't really see anything yet, but here already you see a young patient trying to stand up. You see he's using his hands to get up from the floor, which is because of muscle weakness. So he needs to use this Gowers maneuver in order to stand up because of muscle weakness. You also see that these boys have a weird way of standing, and that's because they lose certain muscles earlier than others. So the way they try to use their function and to keep their function as long as possible is by making adaptations with the muscles they still have. Most patients become wheelchair dependent around the age of 12, and then also upper arm function is reduced. You can see this boy is using gravity to help him raise his arm. You can see this boy is still able to feed himself, but he needs both hands to do this. And eventually patients can use only a few fingers. Obviously, respiratory function is also something for which you need your muscles. So most patients need assisted ventilation around the age of 20. And sadly, patients will die somewhere in their 20s, 30s, or 40s due to respiratory or heart failure. Now, if you hear all these symptoms and how patients lose one after the other function, if you would estimate what the quality of life is that you think Duchenne patients give their life. So from 1, where 1 is death or being close to death, to 10 being ecstatic. So what grade do you think that the patients will give their life? So we see an 8. A 4 as well. That's gonna go all over. I'm just going to wait a little bit to allow other people to also vote. And I think, well, I think the majority seems to agree on 4. While we also have some higher marks. And this is actually interesting because usually when I ask this, the score will be between 2 and 3. So already these people are, the participants, you are higher than that. And also some people score quite high. Well, what I can tell you is that the average that healthy individuals usually score their quality of life is somewhere between 6 and 7. And that's the same that Duchenne patients do. So Duchenne patients rate their quality of life not different from healthy individuals because they manage to cope with losing one function after the other, and they manage to focus on what they can do rather than what they cannot do, which I think is a very good way to look at life in general. So now we're going to look into the protein and the genetics because I need to understand, explain the therapy that we were developing at LUMC over 20 years ago. So I mentioned already, Duchenne patients don't have dystrophin. And in our muscles, dystrophin acts as a shock absorber by connecting the cytoskeleton, the actin, to the connective tissue, the extracellular matrix that surrounds each muscle fibers. So the crucial domains of dystrophin are located at the beginning and the end of the protein, and in the middle you have 24 repeats that are partially redundant. So this is important. The crucial domains are located at the beginning and the end of the protein. What happens in most Duchenne patients is that they miss one or more exons, and you can see here 48 to 50 are missing, and you see that the reading frame is disrupted. So 47 doesn't fit to 51. So what happens during protein translation is that the translation stops somewhere in 51. So you have only the beginning of the protein, and this crucial domain that connects to the extracellular matrix is not there. So this dystrophin is not functional, and this is what you find in Duchenne. Now you can think, well, what would happen if the reading frame would be maintained? And we have an example here, 48, 49, 50, and 51 are missing. You see 47 fits to 52. So the reading frame is not disrupted now, and protein translation can continue from start to finish. Now for most proteins, if you miss a little bit, the protein is not going to be functional. But our dystrophin had the important domain at the beginning and at the end. So now this redundant repeat domain is slightly shorter, and we know it's partially functional. And we know this because this type of mutations where the reading frame is maintained are found in Becker muscular dystrophy. And this is a disease that has a later onset and also a slower progression. Knowing this, what we were thinking is, well, of course, what you would like to do is to allow Duchenne patients to make the normal dystrophin. With current techniques, that is not possible. So then we were thinking, well, can't we use antisense oligonucleotides to try and allow Duchenne patients to make Becker-like dystrophins? And then the question is, well, how would this work? Well, we're looking here at the pre-mRNA. So we have, again, our Duchenne mutation of exon 48 to 50. We know 47 doesn't fit to 51, but we know it does fit to 52. So during the splicing process, when all these exons are linked together, we want to skip or hide exon 51. And this, of course, we can do with antisense oligonucleotides or AONs or ASOs that bind specifically, in this case, to exon 51, restore the reading frame, and now we get a Becker-like protein instead of a non-functional protein. So this is the theory. But when we started working on this in 2000, well, there were AONs or ASOs already, and of course they had to be modified chemically because you have to make them nuclease resistant. We should not induce RNase H, we want to modulate the splicing, and they needed good pharmacokinetic properties. Now in 2000, if you now look, there's so many chemical modifications that it's really difficult to keep track, but in 2000 there were not that many. The one that we used was the 2-methylphosphorothioate, and that was the one that was well, used by most people. There was data about it. We knew it was stable. It was cheap, though, well, cheap is a relative term. It was less expensive than the other options, I should say. And it had good pharmacokinetic properties because it has this phosphorothioate backbone. You can see the sulfur here, which means that it binds to serum protein, which prevents renal clearance. And this is also very similar to the chemistry that Ionis, back then they were called ISIS, they were using to do their clinical trials. So we knew it was already used in humans. So what we did, we designed our AONs targeting exon 51 for a patient from whom we had cultured muscle cells. So these cultured muscle cells we transfected with the AONs targeting exon 51. We did RT-PCR. This is before treatment and after treatment. You see a shorter band. We sequenced it. Exon 51 was skipped as intended. We also did Western blot, no dystrophin before treatment. This is the healthy control. And you see after 16 hours, there's some dystrophin and then increasing levels for up to 2 days. So when we looked at individual muscle fibers before treatment, no green cells, so no dystrophin positive cells. And after treatment, the cells could be stained with a dystrophin antibody that turned them green. So this seemed to work. Now we had a question, because this we used the 2-O-methylphosphorothioate, but then we also tested some other chemistries. And one new chemistry worked really well in the cultured cells, but it had never been tested in humans. So now what do we use? So the previous slide that I showed, that was the 2-O-methylphosphorothioate. So it worked well enough, but we had a new one where we saw more exon skipping. So which one would you use for your clinical development? Would you go for the one where you already have your human data? Or would you go for the new chemistry where you have your, you have no human data, but you know in cells it works better? So I'll again wait a little bit to see what you would do. And again, like there's no right or wrong to these questions, because well, you would do what you would do. Okay, I'll give you some more seconds to log your answer. And also this is interesting because, so whenever I give this lecture to medical students, they will go for the standard oligo where there's human data available. But whenever I give this lecture to an oligonucleotide or chemical background people, they will go for the new chemistry. Well, I think this is one of the topics where, when we started to think about clinical development, we had a lot of discussion and there were pros and cons to both. But in the end, we went with the 2-O-methylphosphorothioate chemistry because we were sure that at least it was tolerated well in humans. And what we didn't want was to use a completely new chemistry that had never been in humans and that might not be safe. It might also not be effective, but I think safety was a more important concern. So now I gave you the example of exon 51 skipping for that one deletion, but of course, patients can have deletions or other mutations all over the gene. So this is a mutation-specific approach. If we skip 51, that applies to 14% of all patients, but that means it also does not apply to 86% of patients. So if we want to increase the number of patients that benefit from exon skipping, we need to develop this for additional exons. So it's a mutation-specific approach, and skipping a specific exon applies to only subgroups. And that meant that we had to explain this, because before we started to develop exon skipping, whatever approach people were using or developing, the mutation wouldn't matter. It would apply to everyone. And now suddenly it mattered what mutation people had. So we had to explain how this worked and why this was. That we were not just saying, well, no, you cannot get this treatment because we don't like you, but because for you it won't work because your mutation is not eligible. So we had to do a lot of patient education, and we did this trying to explain things in simple words, but we even also had a dance where we tried to explain how things worked in dance form. If you're interested, you can click the link. But what's also important when we discussed this with patients was to have realistic expectations. And it's one of the phrases that already came up when I asked how patients can be involved, because this is not a cure. What it will do, it will slow down disease progression. It's not a fully functional dystrophin, and you only intervene at a later time point. So we needed to make sure that patients were aware of this. And then a question again for you, do you think that this is what patients want? Do you think, well, no, they don't want this. They only want a cure because, I mean, it's going to be an invasive therapy. They will have to be injected repeatedly. Yes, they want this, but only if it prevents them from losing ambulation. Or yes, they will want whatever approach slows down disease progression. And again, I see people already start answering. And it's anonymous, so don't worry, I will not be able to track down who answered what. Although I do notice now that Ronald appears to be person number 7 to answer. But I think the majority seems to think that they will want whatever approach slows down disease progression. And of course, look, I'm now asking you, but the important thing is we shouldn't ask ourselves, we shouldn't ask clinicians, we should ask the patients. And this is what the Duchenne patient community in the US, the Parent Project Muscular Dystrophy, did. They did preference studies with caregivers and later also with Duchenne patients to ask, what do patients want? And then it turned out that indeed what the majority of you thought, what patients want is not to lose the next function. Like if they're walking, they want to remain walking. Once they've lost ambulation, they want to be able to use their hands. They want to be able to operate their PlayStation. Once they've lost that, then they want to make sure that the one thumb that they can still use, that they can keep using it because they operate their wheelchair with that. So what they don't want is to lose the next function. So if you can slow down disease progression, that is of course what you're doing. You hope to delay when they lose the next function. So knowing that this is actually something that patients do want, we started the clinical development. And when I say we at LUMC, we started a collaboration with Prosensa, which was a Leiden-based company that were going to do the clinical development. The first thing we did was do a local injection. And it is not because this is what we think would work, because we have 700 different muscles. You need to treat all of them. AON's treatments need repeated injection. This was because we wanted to make sure that in human patients, this worked. And I say human patient because we were allowed to skip the healthy volunteer step, which makes sense because we used an ASO that was already used in humans for other diseases. But also if you skip exon 51 in a human healthy individual, you're going to disrupt the reading frame. So what we're doing in patients is the opposite from what we would test in the healthy volunteers. But because we had to start in patients, that also meant we had to start in children, because when patients are older, a lot of the muscle will have already deteriorated, replaced by fat, replaced by adipose tissue. So we needed relatively young individuals, which meant children. So we wanted to be safe and make sure that local treatment worked. So you can see here a patient. So this is the site where the clinician is supposed to inject. This is an anesthetic cream, and this is the needle that he used to inject. And it's actually an EMG needle that gives a sound when you hit the muscle with the needle, just to make sure that muscle was injected. And you also see here that the foot is fixed for the injection, but also for the biopsy. And that was to make sure that the biopsy was taken from the same place where the muscle was injected, because your foot and your skin, like the muscle and the skin, move independently. So you can do a small temporary tattoo to say this is where I injected, but if the foot was one way when you injected and the other way when you took the biopsy, then you will still miss where you injected. So 4 patients were treated. A muscle biopsy was taken 1 month after, and I show you here a healthy control muscle stained for dystrophin, and the Duchenne patients stained for dystrophin without treatment, and the 4 patients who were treated. And it's quite clear that dystrophin was restored in many muscle fibers that were locally treated. But I mentioned already, local treatment is nice for proof of concept, but this is not a viable solution for Duchenne, because almost all muscles are affected and you need to do repeated treatment with these AONs. So you need systemic delivery. Now, I mentioned already the phosphorothioate prevents renal clearance, so you get longer and better bioavailability, but the ASOs will be primarily taken up by the liver. What we knew from other ongoing trials in other diseases is that uptake in muscle is poor. So that's a bit of a challenge. Still, we said together with Prosensa, let's just see what happens. Let's just try in mouse. So we tried in wild type mouse, but we also tried in the MDX mouse. So this is a spontaneous mutation in mouse dystrophin exon 23. So this mouse doesn't produce dystrophin, it has dystrophic muscle. The phenotype is milder than in humans, but it has no dystrophin. If you skip 23, dystrophin will be restored. So we could use it to at least test proof of concept and to see whether systemic delivery was an option. So what I'm showing here is that if we did intravenous delivery and then we looked where the AON ended up, it ended up primarily in the liver, as we knew, in both wild type and MDX mouse. But then we looked at the muscles, we could see that there was much more AON ending up in the muscle of the MDX mouse than the muscle of the wild type mouse. And that is probably because the wild type muscle is not leaky and not damaged, it's healthy, but the MDX mouse muscle is leaky. And we know there's things leaking out, so there's muscle damage markers like creatine kinase that leak into the bloodstream. But we also think, well, if things can go out, things can go in. So the delivery to the dystrophic muscle is probably about 10-fold easier in the mouse than the delivery to the healthy muscle. So where in healthy muscle it's really difficult to deliver, it's at least possible a little bit in the dystrophic mouse. We also tested different delivery routes and again looked how much goes to muscle, kidney, and liver. And we tested intravenous, subcutaneous, and intraperitoneal. I'm not going to discuss intraperitoneal. You can do it in mice, not so realistic in humans. Intravenous and subcutaneous, so they both work. As you can see, you get a little bit in muscle, you get a lot in liver and kidney, especially if you do intravenous delivery. So now I'm going to ask you again, what route of administration would you choose for your clinical development? Would you go for IV because you get more in the muscle if you do IV than if you do subcutaneous? Or would you go for subcutaneous? Because if you, again, remember the picture from the previous one, if you do subcutaneous, you get slightly less in muscle, but a lot less in the liver and the kidney. So which would you choose? Also thinking that subcutaneous is less invasive than intravenous delivery. Again, I'm gonna wait a little bit, but it seems that the majority of people seems to be inclined to go for the subcutaneous treatment, which is also the one that Prosensa and say the field selected. So then, if you can now treat patients systemically and treat all their muscles, then you of course also expect that they will decline slower and that you can measure this in clinical trials, because in a clinical trial you will need to show, is there an effect? And again, this is also something that came up in the things, how can patients help with treatment development? You need outcome measures. So you need outcome measures that measure something that's relevant to the patient, and then you need to show that the treated patients do better than the placebo-treated patients. So you need all this. So when we were at the stage that the clinical trial started, it's 2007. And then I can tell you that in 2007, the TreatMD Network of Excellence was an EU-funded network that was started to make the neuromuscular field clinical trial ready. So we were ready for trials to happen with our exon skipping compound, but the field was not clinical trial ready, because all these things that you need, like outcome measures, but also registries of where the patients are and what mutations they have, etc., standards of care, they were not there. So TreatMD still exists, so there's a lot more now than in 2007, but it's definitely work in progress. And yeah, so we needed to decide, well, there are no outcome measures, so what are we going to do? So what we were not going to do is wait and develop outcome measures, because this can take 3, 4, 5, 6, 7 years, if not more. So trials were initiated, and Prosensa licensed the exon skipping drug to GSK after the first dose escalation study. And then GSK said, in parallel, we're going to design 3 different studies, all with a placebo group, a dose regimen, a dose comparison, and an efficacy study. So all these studies had an open label extension phase at the end. So even if you were in the placebo group, you could then get the treatment after the trial period was done. And lacking an outcome measure for Duchenne, the 6-minute walk test was selected. And the 6-minute walk test is what it says on the tin. You walk as a little boy in the hospital hallway for 6 minutes as quick as you can, and then they measure the distance you're able to walk. So this is an endurance test from the cardiovascular field. And the idea is, of course, that, well, if your muscle disease progresses, with time, you will be able to walk less in 6 minutes. So we can use this to see whether there's a slower disease progression. So before going into the functional effects, I'm first going to go into the side effects. These happened for all the different clinical trials. So I showed them once, but be aware they happened everywhere. And the one that was most burdensome was the local injection site reactions. So if patients were treated for a few weeks, there was no problem. But they were treated weekly, and even if they rotated the infusion site, at some point patients started to develop ulceration, discoloration, there was loss of subcutaneous fat. And for some patients, this started happening after 6 months, for some after 2 years, but eventually it happened for all patients if they were treated long enough. We now know that this happens if you do frequent subcutaneous delivery of phosphatidylate AONs. When we made this decision that this was more patient-friendly, we didn't know. Like, had we known, we would of course have said, let's go for intravenous delivery, because you don't have these injection site reactions. But we didn't know. So in hindsight, the choice was wrong. But yeah, because we didn't know, the choice back then was correct. What we also saw in patients was proteinuria, but during treatment breaks, this was reversible, and it was also subclinical. And in a few patients, there was thrombocytopenia, but no one had any bleeding incidents. So I'm showing you an example of the skin reactions, and you see that, well, they're relatively mild. So this is some discoloration. Here, some of the subcutaneous fat is gone, but that could also be quite severe. So this really, there's scar tissue formation, and this is not reversible. So this is really quite severe. And also, like, even the discoloration, some boys didn't care, but some were really conscientious about it. So it was not a good thing. In hindsight, intravenous would have been the better choice. But again, like I said, we did not know. Now, I mentioned, the first study that was done systemically was a dose escalation study. And what we could show is if you did systemic treatment, patients started to make dystrophin. So then, after 12 weeks of treatment, patients were offered the choice to go into an open label study and to receive the treatment for however long they desired, weekly. And all 12 patients wanted this. And well, got weekly treatment for 1.5 years, meaning 1.5 years, no holidays, going to the hospital every week, a day of work for parents, a day of school for the children. And of course, a day of school might be nice once every so often, but if it happens every week, it becomes really a burden. So then they changed the regimen to 8 weeks on and 4 weeks off, and they said, well, let's just see how it works, and it works similarly. So there were 12 patients in this trial, but you see data from only 10. And that is because 2 of the patients were non-ambulant, and obviously if you're non-ambulant, you cannot do the 6-minute walk test. The 2 who had the lowest baseline lost ambulation in about a year. The other 8 were stable for 3.5 years. So they were stable when they received weekly treatment, and they continued to be stable when they received the on-off regimen. So based on this data, do you think this drug works? No, yes, I need more information, or I do not know. Most people want more information. I would agree. It's an open label study. It's nice, it looks promising, but ideally we want a placebo-controlled trial. So now I show you the dose regimen study. So in blue we have placebo. In red we have patients who were treated every other week, and in green we have patients who were treated every week. The baseline of the 6-minute walk test is set to 0. So if the line declines, patients were walking less. And if it increases, patients were walking more. And then you see after a year that the patients with the placebo group were walking about 30 meters less than baseline, while the patients in the 2 different regimens were either walking more or about the same as the baseline. So now, having seen this, do you think this drug works? No, yes, I want even more information, or I do not know. want more information, but some seem to now think it works, some think it doesn't work. I think it's... Most people seem to want more information. So we have the other dose comparison study. So here, what was done, so this is a trial for half a year. So what you see is that in blue, the patients lose about 15 meters if they're not treated, and they were given half the dose that the patients you've seen so far were given, which was suboptimal. But what I'm asking you, the dose that was used in all the trials, the 6 milligram per kilogram in red, here you see that the patients who were treated compared to the baseline showed an improvement. So now, based on this, do you think this drug at the optimal dose works? Yes, no, I need more information, or I do not know. Again, gonna wait. People still want more information, but some also seem to think it works. I think that the audience might be somewhat enriched for people who have already heard the story. But I think so people want more information, but also people seem to think it works. And you could see at the beginning, there were some people that said no, there's no one who says no. So people want more information. I think so if you look at the situation that the field was in when we saw the 3 different clinical trial results, we thought, okay, this drug seems to work, and we now need to show with our phase 3 clinical trial that it indeed does work. And this is what the phase 3 trial showed. So in blue, placebo, in green, the treated group, and there's different things you can see. So the first thing you can see is that both treated and placebo go down, and also that they go down a lot more than the previous slides showed in 1 year. But you can also see, well, the green line, the treated group does better, but very little. And this is, of course, this was 11 meters more. This is not statistically significant and also not clinical relevant. So now what do you think? Do you think this drug doesn't work? It does work. You want still more information, or you're no longer sure what to think? We lost the yeses as expected. And I think so. I don't know what to think anymore. That was, of course, also what, as a field, we were like, well, it all looks so nice and promising, and now suddenly it doesn't work. Some people indeed said, well, then it doesn't work, but some people also said, well, we need more information. So what we thought about the 6-minute walk test before the trial started with Duchenne was, patients walk a certain distance, and then every year they lose a little bit until they go from 350 to 0. And what we know now is that is not how it works. So young patients can be stable even for 2 or 3 years. So patients who are less than 7 years old, they can be stable. They can even increase a bit when they're growing. And patients who are older, they start to decline. But you can also see, like here, they decline until 200. But what we also know, you can either walk about 150, 200 meters, or you stop walking altogether. So then you see a very rapid decline where the last few muscle fibers that allowed ambulation are lost, and patients then stop walking altogether. So this linear thing that we thought of how the 6-minute walk test worked, that was not really reality. And what we then did, or what then happened, GSK stopped the development of drisapersen. So Prosensa got back all the data. Prosensa was acquired by BioMarin, and BioMarin did additional analysis. Because what they saw was that the patients who were in the phase 3 trial had a more advanced disease. So I said already, the patients, the drop that you see is much more than what you could see in the other 2 trials. And that was because these patients were older and in a more advanced stage. Now, if you looked in the subgroup that would be allowed to be in the phase 2 trials with the inclusion criteria, then there was a treatment response that was very similar to the one in the 2 earlier trials. So if you only focused on this subgroup, where probably you're able to show a treatment effect because there is disease progression, so it's not stable whether you treat or not, and it's also not so far advanced that there's beyond the point of no return. If you are in that sweet spot, then you can show a treatment response. In that group, there was a treatment response shown. Now, if you know this now, so how this 6-minute walk test works in Duchenne, do you now think this drug works? Yes, no, or I don't know whether it works. 50-50. We're losing audience. We're losing audience. Oh, no, more votes. So people don't know. And I think, so I'm with the people that don't know. I don't know whether this drug works. It might work, it might not work, but I think the problem is that the results are inconclusive. What BioMarin did is that, well, this younger subgroup shows a response. They combined the data of all the patients who are in this subgroup that showed the response and the phase 2 data. They combined everything and they applied for approval with FDA and EMA. FDA denied approval because they said, well, you did a lot of post hoc analyses and also there's these side effects that we don't really like. EMA, the meeting happened behind closed door that they had, and before EMA gave an approval or not, BioMarin withdrew the application, which probably meant that that discussion that they had with EMA didn't go too well, because also 2 days later they announced that they were just going to stop the development altogether of the development of this ASO, because they said we need better compounds. So let's see, this is, yeah, so they stopped the development. They said, we need better compounds. We need something that works better and is safer. So this was, as you can see, this was in 2016. And as of this year, there's a new clinical trial ongoing with a compound developed by BioMarin, so the next generation. But that meant it took them 9 years to go from this trial to towards a new development. So it's a bit of a sad story because, well, why did it not go right? Because we lacked the proper outcome measures. And why did we lack outcome measures? Because as a field, we started to talk to the regulators too late. So we didn't realize we need outcome measures. And then, well, you can say, let's use the 6-minute walk test. But if you don't have the natural history data that I explained that young patients can improve and then they're stable and then they start to decline, if you don't have any idea of that, then it's impossible to design an optimal trial. And if you design a suboptimal trial, then, well, what can happen is that you have results that you don't know whether the treatment works or not. Again, of course, if the treatment effect is very impressive, like the nusinersen, where patients gain skills that they otherwise would never have gained, it's easy. And even with suboptimal trial design, you will see that your treatment works. But the treatment effect was always going to be minor, so it's much more important to have good outcome measures. So the situation has changed, so we have an ongoing dialogue now with academics, patients, and regulators. We've developed new outcome measures where, as someone suggested already in the very first question that I asked, we started with the patients. We asked the ambulant patients which functions are important. We asked the non-ambulant patients which functions are important. And then based on that, we made the outcome measures so that we know now if these things decline, something is lost that matters to the patient. So the future trials will be and are better. So where are we now as a field? There's 4 AONs approved, so the 2-methylphosphorothioates stopped. 4 AONs approved the phosphorodiamidate morpholino or PMO chemistry. So they have no phosphorothioate backbone, which means that their bioavailability is very limited, which also means that probably you get only very minimal effect on dystrophin restoration. So these 4 drugs are approved because there's usually around 1 or less than 1% of dystrophin restored after treating with weekly intravenous infusions with very high doses. So the doses are 30 to 80 milligram per kilogram per week. What's not yet been shown is whether this also slows down disease progression. So it's only based on dystrophin restoration. And clinical trials to ascertain this are ongoing. What is obvious is that there's room for improvement, because even if 1% is going to slow down disease progression, 10% is going to slow down disease progression more. So we need to improve this. So what we're working on in the group here in Leiden is to improve Duchenne exon skipping and delivery, have better model systems, have natural history studies also for our animal model system, improve the muscle quality. And so we're doing lots of this work. And don't worry, I'm not going to talk about all of this, but just to give you an idea that based on where we are now as a field, we're trying to improve. So finally, the acknowledgement. So people involved in the group, you see here a picture of the group. This was just before the pandemic, and usually I show a newer picture, but this one is nicer because these were the people who were actually involved in the earlier stage work that I showed here of the preclinical and the clinical development. Of course, collaboration with the Department of Neurology, because we are mouse doctors, we're not allowed to treat patients. So for that, we need the clinicians, and also collaboration with Procenta and BioMarin along the way, and funding from a lot of organizations. And I want to mention specifically the Duchenne Parent Project, which is a patient organization that gave the very first grant to start this work. So again, another way that patients can be involved in therapy development. So with that, I will try to stop sharing and happy to answer any questions you may have.
Yeah, thanks a lot, Annemieke. So I'm trying to do it like this. So I'm using Annemieke, her microphone. So I kind of assume that this works. Quickly looking at Erin. Yes, it works. Okay. If not, please let me know. Then I will change it. So Annemieke, thanks a lot for this excellent presentation. I think it's always very important to have patience in your research. So I think it was your very first question that you asked, like how can people, how can patients or family members of patients contribute to your research? So can you tell us how you collaborate with patients?
Yeah.
Is it only asking questions and questionnaires and looking for new outcome measures, or is it also in writing grants?
Yes, so very good. So I think so the first way they collaborated, or that we started collaborating, was really because a grant application was given by the Dutch Duchenne Parent Project. So that is how it started. Then we also started to have interactions with the patient organizations and to work with them. And I think so. What I do is a lot of patient education, so explaining to patients how things work, and then see what their feedback is. What is difficult, so patients are very much well able to say, well, this is the burden of my disease. This is the 24/7 living with the disease. And I think that is something that clinicians may be less aware of, because we're researchers. We don't see many patients unless we go to patient days. The clinicians will see the patients because they will come to their clinic. But that 1 hour every so often is, of course, not the day-to-day living with the disease. So I think if you go to patient meetings, that is what you will hear. What is it like to live daily with the disease? So I think that is not really a collaboration, but just go, if you work on a disease, go to a patient meeting, talk to the patients, talk to the families, and see what it's like to live with the disease, because I think there's nothing more motivating than that. So that is one way that we're collaborating. I think, well, definitely for the outcome measures, I think as Treat-NMD, that is one aspect that we've been involved in. And I think so it would be nice if patients would say, well, you should, like, I have a great idea to improve exon skipping, but that's not realistic, because you need a PhD probably, or at least to understand genetics. So that is less how we collaborate with them, but we do collaborate with them to see, well, what are the challenges? And I think, so how can we improve exon skipping? The most important thing they want is not to have weekly intravenous infusions. So, okay, so we need to take that into account. So it's better to have something that you need once every 6 weeks. That is a better solution than having something that you need every week and then gives a better result. So these are the ways to talking and listening. But we're also, like, that's not part of the exon skipping work, but just say, as a field, having this dialogue So translate and interact between the regulators, patients, the clinicians, make sure that there is this ongoing dialogue to make everyone aware what is what, which is like maybe not core business as a researcher, but you need these tools. And I think one other aspect, if you're a researcher, it's good to talk to the patients, because it will also make you aware, like, okay, so I'm now doing this in my mouse or in my cultured cells or in my organoid, but in the end, I need to test it in a patient. How will I test this? And then maybe you're not the person developing it, but you need to be aware that someone needs to do it. So I think communicating with patients is probably the executive summary of the long monologue I just gave.
Yeah, thanks. Yeah, I fully agree on that. Maybe a very small follow-up question. So I think, of course, talking to patients, and this is super communication. This is super important, both ways, I think. So I think what you see more often is that you need patients for grant applications. So can you give some advice how patients can contribute to grant applications? Because I think what I hear a lot from patients and patient organizations, that a researcher writes a grant, and then one day before the grant deadline, oh shit, we need a patient organization. Can you please sign this document? The patients on the grant. So how would you approach this?
So I think this is, of course, like, you need to approach them earlier, and ideally you already know them and you have already talked with them. So just the idea, like, and I think also that you cannot expect that patients have the level of knowledge that you have, because like, they don't have time to work on this the whole day. They have other things, they have their other work, and they usually also have their disease to cope with. So you need to be able to explain to them in simple terms what you are doing and what you expect and why you think this is important, because then they can say, okay, indeed, this is important or it's not important. And I think if you want them to actively contribute, then you need to educate them in how things work. And it's okay if they don't understand how transcriptomics work and how that's way too much detail. But if they understand the gist of things, then I think that is that's important. And I mean, again, it's not very likely that they're going to say, well, this is the best way to develop your guide RNA. Like, that's not realistic. But if they understand, like, this is what you want to achieve, and that is something that I would be open to, that is something where they can, of course. And also, like, if you see in your animal model, well, it seems to affect these symptoms, but these symptoms are still the same, that's when you can ask them, like, would that still be relevant to you? So I think it's again, it's a dialogue, but it starts with explaining to them. So in simple terms, as briefly as you can, how does this work and what is expected? And then I think also, like, it's nice if they have this letter of support, but ideally at some point you also invite them and say, look, these are the results and this is what we achieved, and well, what do you think?
Yeah, of course, that would be next. So in my eyes, it's always, we are researchers, we are in the lab, and we are very happy with our Western blot and our PCR. So it's a very nice result. But also for the patients, because it was also in your webinar, I think one of the things that they want a cure, right?
Yep.
They come to the researchers, to the physicians, and they want, can you develop a cure for me? So how difficult is it to explain what you do in the lab? And that, you showed the example that the first clinical trial was in 2007. We are now in 2025. So it's quite a lengthy process. So how do you explain this to the patient that this takes a lot of time?
I think what's important is you explain how things work. So they don't need to understand the Western blot. They don't, like, the PCR doesn't really, like, just show this is why it works. Look, protein is missing because there is no band. Now there is a band, the protein is back. That is the level that they need to understand. And like, when you present to researchers, you will say, oh, look, it works because of this. You don't need that level of convincing. Just look, it works because we see this. But then also, like, explain which steps are needed. So what do you need? You go from cells to animals to phase. So when you found that something works in cells, you can be super happy. But of course, there's a lot extra steps. So that is important. Like this is the roadmap, this is the route, this is where we are now, and these are all the next steps that we still need to take. And these take time, but also the answer may be no. So it might stop after the next stage because, well, it works in cells, but it doesn't work in mice. So I think that is important to make them aware that it's not just that it takes time, but also that every next step is more complex. So that means that every next step can fail. And I think it's better to be honest about this than to overpromise. And also, so of course, patients will very often ask, okay, so when? You have your cell results, you have your Western blot, you're super proud. So when can I use this? Just don't give them an answer, because yeah, if all goes well, 5 years, they will mark it in their agenda and they will come back to you in 5 years and say that, so where is it? Yeah, well, it didn't go beyond the mouse because, well, it worked in my organoids, it worked in zebrafish, but then didn't work in mouse, or it killed all the rats. Like, so just don't give them an answer and say, look, we don't know. We're at the first step, there's new steps, this will take time, and each of these steps can fail. Because it's very nice to give the patients hope, but if you give them false hope, at some point they're going to be incredibly disappointed, and that is way worse than the level of hope that they have if you give them a wrong answer.
Also fully agree on this answer. So again, there are quite a few questions in the chat. I will read them. I will ask them. I have one more, because I was a little bit surprised by the quality of life. That the quality of life of the Duchenne patients was similar as healthy individuals. So how come? And what about the Becker patients? Because you would say that. And this, so what about the Becker patient? And maybe also, is there like a difference depending on the disease stage?
Yeah, so I think so. What you see in Duchenne patients is that they have a quality of life, and then they lose ambulation, and then it will go down, but then it will go back up. And then they lose the next step, it will go down, it will go back up. But the dips, each time they lose the next function, the dips get lower. So they even adapt to losing a function. So why is it not different from us? I think has more to say about us than about Duchenne patients, because we probably mainly look at all these things that we don't have, all the nice things that Ronald has, the stroopwafel that Aaron has at the moment. Like, everyone else has things that I want. But Duchenne patients, they can look at that and see, well, they can walk. I used to walk, I cannot walk. Well, that's just going to make you depressed. So what they learn is, let's focus on the things that I still can do and enjoy that, because tomorrow I might no longer be able to do it. And I think that's, of course, a different mindset. And I think Becker patients, there's a lot less research in Becker patients, which I think also should improve. But I think Becker patients, the progression is slower. I think their quality of life might actually be worse than in Duchenne, because like they have, well, and also like they're adults sometimes when they have the disease. So it's a different perspective. But I think so, like, and I mean, I could completely understand if Duchenne patients had a poor quality of life, like they deserve to say it's a 2. But the fact that they don't, I think, should motivate all of us to be more appreciative of all the functions that we do have, rather than focusing on all these little things that frustrate and bother us. And I try to live by that, but I don't always succeed.
Also fully agree on this answer. So we agree on a lot of things. So moving to a question from Eric. It's about the clinical trials. So how did they come up with the dose level and dose frequency for the clinical trials?
So there was preclinical studies in the mouse. Of course, because the phosphatidylate improves the bioavailability, there's this scaling that you do because the surface area to circulation area is different in mice from human. So they saw what is needed in mouse, and then they did PK, PD, PKPM modeling. Don't ask me the details because I'm not a pharmacologist. But they managed to say, okay, this is the dose that we should use in humans. And then they did a dose escalation study starting with the low dose, where they said, well, for sure this is going to be safe. And then a higher dose when that turned out to be safe, and a higher dose and a higher dose. And it turned out that the highest dose, the 6 milligram per kilogram per week, showed most dystrophin increase. So that's how they ended up with that dose. But I think this is also important. So you need to do a lot of boring preclinical studies before you can do your clinical trial, because you do need to indeed know. So we tested tons of different things. What if we do daily injections? What if we do every 2 weeks? Like all these different things you have to check and then see what is optimal and correct also to the correct for the human versus mouse differences.
Thanks. A few short answers from Jawad. Great talk, Annemieke. Agree on that. So what sequences of motifs have you found to be important for exon skipping efficiency?
Okay.
So does
There's several papers about that. So I would say PubMed, Aartsma Rus, guidelines. I think there's one on AON design or ASO design that you can look up, and probably Aaron or Ronald can also put them in some documentation. So we looked at AONs that worked and ones that didn't work, and then we compared them, and then we saw that there were indeed some things that predict whether they work or not. So there's a lot of work on that, and I would say just Google and PubMed, and you should be able to find a lot of these papers.
Yeah, I think there's a document that we use for every new ASO development. We use a document from Annemieke to design our new ASO, so you can easily find it online.
And if you don't have access, email me and I'll send you the PDF.
Yes, and then the follow-up question from that, short answer, please. Does proximity or overlap to splice sites play a major role?
Yeah, so interesting question. So all the approved AONs for Duchenne are not targeting the splice sites. They're exon internal, so they're within the exon. Whether that's something specific for dystrophin exons, I don't know. Dystrophin introns are huge, so it might be that there's more internal motifs that are involved in splicing. What we did see, so AONs targeting exonic regions seem to work better than those that target splice sites. For dystrophin, whether that applies to other exons, I don't know. But also, if you have the choice, the earlier in the exon, so the more to the beginning of the exon, the more likely it is that your AON will be effective. Targeting the splice sites probably work less well because the GC percentage will go down, and also the splicing factors like the U1 and the U2 and the U2AF, they will probably bind with much more affinity, and they might be able to kick off your AONs, while all the other splicing factors bind with lower affinity, so the AON may win and have an effect.
Thanks a lot. One final question from Feng. So what about conjugates? So you can, because I think I've seen a lot of presentations in the last few years. People are conjugating the ASOs and see a way better uptake. So what's your perspective on that?
So we've done work also with the transferrin receptor 1 antibody conjugates. It works really well. There will be a poster about this at OTS from Tiberius Stam, so you can see for yourself how well it works. So indeed it works, and this is also in clinical trial for Duchenne. But the challenge, of course, if you have these conjugates, and so if they're antibodies, you can get antibodies to the antibodies, and you can get infusion reaction. And also, if you target a transferrin receptor, then of course your iron transport might also get influenced. So it seems to work, but whether it will make it all the way to clinical application, that we don't know yet, because the trials are ongoing. And there's other conjugates, so you can use arginine-rich conjugates. That leads to kidney toxicity, and those trials have been stopped in Duchenne.
Thanks a lot. So I give the word back to Erin.
Great. Well, thank you, Annemieke. That was a great talk as always. It's always informative. It's nice when we can ask the speaker anything, and Annemieke is happy to answer. We have a few webinars coming up, all sorts of different topics. So some on strategic IP for oligonucleotides. We have one coming up on a database that really gives a good idea of the neurological oligonucleotide landscape. financing the future of individualized medicine, and then that brings us right up to the meeting. So do take a look on the webinars page of the OTS. And again, we just want to thank Anamika one more time for being such a support of the webinar series and always giving us great talks.
Thank you.